Hello everyone, today's topic is T-cell development. What we will learn in this section? So we will take these two questions. The first one, what is MHC restriction? And the second one is what is the non-self-reactive T-cell, or what is the self-tolerance? So this section we learn the T-cell development. So we know all the cells is from bone marrow. So the T-cell also precursor from bone marrow and move to thymus. So these cells grow and mature in thymus. So we call this lymphocyte is T-cell. So do you remember what is the primary lymph organs? So they are the bone marrow and thymus, right? The primary or central lymph organ, the function is to mature the T-cell or the development of lymphocytes. And according to the CD4 or CD8, these two molecules expression we name different stage of T-cell is double negative T-cell, double positive T-cell, or single positive T-cell. So at the beginning, at the early stage for T-cell development, this T-cell only start from T-cell expression, but they don't express CD4 or CD8. These two molecules, so these cells we call it double negative T-cell. Later, after that, in the middle stage, the T-cell express both CD4 and CD8, these two molecules, and we call it double positive T-cell. And later, this double positive T-cell express only CD4 or CD8. So this stage, we call this T-cell as single positive T-cell. So let's see the stage or the development of T-cell. So this is the structure for thymus. So the T-cell just from bone marrow and come into thymus, move from cortex and into the cortical medullary junction, and later they will go to the medulla, and during this course they finish their development. So this show you the T-cell in the electric microscope, and this also the structure for the thymus under the light microscope. So the T-cell from bone marrow come into thymus, and they move from cortex to the medulla in the thymus. They finish their selection. So here, I always introduce the T-cell will pass two strict examinations. So later you will know how strict of these two examinations. So let's take this question. If we have 100 students in our class, and we want to pass one examination, how do you think, how many students pass this examination and is strict? So if we have 100 students in one class, only half, 50 students pass this examination, or only 20 students pass this examination. Do you think this examination is strict or not? So let's see the T-cell development. How many T-cell can pass these two examinations? First, the positive selection, the first examination. So let's see. The T-cell now from double negative T-cell into the double positive T-cell, both CD4 and CD8 expression, right? And let's see the epithelial cell in the thymus. They express both MHC1 molecule and MHC2 molecule. So now the T-cell, the double positive T-cell, binding with MHC, this is by chance. So now, for example, this T-cell can recognize this class 1 molecule. So this TCR recognizes this MHC1 molecule, and this is a double positive T-cell, right? So the CD8, this molecule, binding the alpha-3 domain of class 1 molecule, right? So now the CD8 molecule receive a positive signal. So the CD8 molecule will remain on this T-cell. And CD4 molecule doesn't receive signal, right? So this CD4 molecule will disappear. So now this T cell, this double positive T cell, it has both CD8 or CD4, these two molecules, but this T cell meet the class 1 molecule. So the CD8 molecule remained and the CD4 molecule disappear. By chance, if this T cell can recognize this class 2 molecule, and this CD4 molecule will bind the beta 2 domain of class 2 molecule, right? So this CD4 molecule remained and the CD8 molecule disappear. So this is the T cell can recognize the MHC molecule, and this T cell become a single positive T cell. And other T cells, this TCR cannot recognize the MHC molecule. So this kind of T cell cannot survive in thymus, and this T cell will die, apoptosis. So this cause, we call it positive selection. So that's C. For positive selection, the TCR must have a very good moderate affinity with MHC molecule. So this T cell can recognize the self-MHC, can recognize this MHC molecule, and this T cell will survive. But those T cells, their TCR cannot recognize MHC molecule, so this T cell cannot survive. So this positive selection gave this T cell will get the self-MHC restriction. So previous, you also see the TCR recognize the peptide expressed on MHC, right? And I cite for our body, the TCR only recognize our own self-MHC. We cannot recognize other people's MHC molecule because we have this T cell, the positive selection. So the positive selection make the T cell have the self-MHC restriction. After positive selection, now the single positive T cell move from cortex to mandala, and now in mandala of thymus, the dendritic cell or macrophage or their epithelial cell can express our own antigen. So that's C. They have this gene, tissue restriction gene. So that's C. The lymphocytes, mammary gland, so almost all our organs in our body here can express in thymus. Our heart here, right? Our heart and our tongue, our kidney, our pancreas, all our organs in our body. So this antigen can present in thymus. So now that's C. This dendritic cell expresses peptide. So if this T cell can recognize this peptide, what does it mean? That means this T cell can recognize our own antigen, right? So do you want this cell survive? So no, of course no. So if this cell can survive, that means if this cell pass this examination is not strict, and this T cell will come out. Maybe this T cell can recognize our pancreas or our kidney and come out, it is dangerous. This T cell is the autoreactive T cell and make us have the autoimmunity. So this T cell can recognize our own antigen, cannot survive. So they will die. So this is the negative selection for T cell in the thymus, and we call it this T cell get the self-tolerance. So this is the second examination for T cell development. So now we know the T cell development in the thymus, and they passed two examinations, right? So this examination is very strict. So now I give you the answer. So if we have 100 students, or if we have 100 T cells come into thymus, so after these two examinations, positive selection and negative selection, only 2 or 4 students or only 2 or 4 T cells can pass these two examinations. Okay, so we have very, very strict examinations for T cell development. So let's see what is a positive selection. So positive selection ensures MHC restriction. The positive selection permits the survival of only those T cells whose TCR are capable of recognition self-MHC molecule. So after that, the T cell got the self-MHC restriction. And the negative selection ensures T cell has the self-tolerance. The induction of death in lymphocytes bearing receptor that react too strongly with self-antigen. So negative selection is an extremely important factor in generating a primary T cell. So this T cell is self-tolerance. So we don't have self-reactive T cell in our body. If we have it, that is dangerous. So okay, this class we learned the T cell development and we know the T cell passed 2 strict examinations. So please know what is it.